of what ecology is. Just like in your own words. And I know I'll just call people randomly. Like how about that? Yeah.
It's a study of population change over time in regards to biological factors, basically.
For sure. Definitely. You know, ecology basically, like in its simplest sense, is like you're studying organisms as they interact with other organisms and as they interact with the environment. And when you think about like the ecosystem and like environmental factors, like there's so many different things that can affect how organisms behave, how they reproduce, how they develop, what genes they express, you know, things like temperature, things like pH of soil, and things like water content, natural storms. All of these different things are affecting how these organisms develop and how these organisms live. And as a result, you see this big variation among individuals in a population. And that's why when you're an ecologist, that's why stats become so important, because stats help you understand and kind of make sense of this extreme variation in nature. And that's partly why like ecology and statistics kind of go hand in hand. So this lab is really like you're generating data, but you're also looking at that data from like an ecologist lens and kind of like looking at the variation in statistics and just trying to make sense of it all in terms of like a biological process that you're looking at. So ecologists, we use these stats to better understand what these could mean in nature and like to help explain things that you see in nature. So we have a couple of words that I'm sure you guys are all familiar with. Who can give me the definition of mean? For stats. Yes.
Sum of all your, I guess, data points and divided by the square root.
Absolutely, yeah, that's what we have right here. So x1 being, or xi being an individual point, so you add all your individual points divided by your total number, which is n. That's the mean, or the average. What about variance? You can tell me what variance is, and like, I guess like what that means in your
How far data points range from the mean?
Pretty much, yeah. So it's the sum of, so xi again being like an individual data point minus your mean divided by your total sample is minus 1. So it's the sum of every individual point and how far that is away from the mean. So it's basically how far your data points lie from the mean. So what about standard deviation? It's similar to variance. So the difference, standard deviation is the average deviation for an individual sample. So variance is kind of like the total amount that is varying, standard deviation being the average amount each sample is varying from the mean. And these are all relative, especially standard deviation. So like if your mean is 100 and your standard deviation is 3, like that's relatively small. If your mean is 10 and your standard deviation is 3, even though standard deviation value is the same, that's much higher. So it's like 30% versus 3% of your mean, you know. So this will vary. Standard deviation is a good way to see like how precise your data points are. So if they're all clustered around the mean, then your standard deviations will be much smaller. Likewise, if they're very far apart from the mean, then you're going to have a larger standard deviation, and that's going to tell you about how your data is spread out and like how much variance you see in the data. So what about the R squared, or coefficient of determination? This one might be the trickiest. So this one's probably the most useful for comparing between data sets because it kind of standardizes it. The way to think of it, like you're almost comparing apples and oranges when you use the coefficient of determination. So it's essentially the variance you have in common over the total variance, and there's equations for this that are generated on Excel. You probably won't ever have to like do this by hand because we have algorithms for it. But it pretty much gives you a value of between 0 and 1 I think of it as a percentage where 1 is 100%. So if you get an R-squared value of 1, that means your two variables are associated perfectly. They align perfectly, which you never really see in actual data sets. But the point being, the closer you are to a value of 1, the stronger indication that your data points are related. So a common misconception that a lot of people have is when they look at coefficient of correlation, they're thinking that the two data points are like one is causing the other, which is not always the case. So for instance, if you're looking at like time on your x-axis and your carbon decay rate on your y-axis, if you see a standard an R-squared value of like 0.9, you could say, well, time is causing a decrease in decay rate. But you can't really say that for certain because maybe it's not just time, maybe it's getting colder too. Like maybe there's temperature, pH involved. Like you can say that these two variables are associated, but you can't say that one is causing the other. Does that make sense, everyone? So that's a really important point. So that classic saying correlation does not mean causation. So that's kind of what this test is looking at. It's looking at these variables associated together, but it doesn't mean that one variable is causing the other variable. So then we also drew a histogram. A histogram would have boxes, but this is the general idea of a histogram. So this is what's called a Gaussian curve, and this will come up a lot in biology, mainly because you see this in a ton of data sets. So you see this in so many different human height, for instance. Like there's an average, so x with a bar on it, this being the mean or the average. You see like the most numbers of individuals in a population are going to be at the mean. And as you get further, so this being +1 standard deviation, +2, +3, +4, as you get further away from the mean in both directions, you see that frequency go down. So you see less individuals with those kind of values. Likewise, you can have some that are even like 5 or 6 or 7 standard deviations away from the mean. Those would be called outliers. So you do see them, but they're not really well represented in the population. So again, what you have is this highest frequency right around the mean, and then it kind of dips off in both directions. So this is the most common one. This is the standard distribution. A lot of the statistical tests that you'll learn about assume that your data set looks like this. And you'll see as you go on, this isn't always the case, and sometimes you have to like change your assumptions depending on like what test you're going to run. Sometimes you have to pick, this would be a parametric distribution, so sometimes you have to pick non-parametric tests, ones that don't assume this distribution. So for instance, you can have other versions of this where you have kind of like two tails. So something like that's going on. So you'll have like two peaks. So you have two peaks in it. And sometimes your data is not evenly distributed, so that way you have to kind of change where you're looking at. Obviously the average would be different, but you can have these skewed left, you can have them skewed right, depending on what environment what environment it is. So let's say we're looking at like snail size length, snail size. So if this is snail size, if they get too big, maybe they're too obvious to predators and they always get eaten. So maybe you'll have like a drop off toward the end and you won't see large snails because they get selected against the environment. Likewise, maybe if they're too small, they get eaten too quickly and they don't grow to full size. So I feel like that might be throwing too much at you, but just think that like you're not always going to see a standard distribution. The environment will have certain effects that will naturally select against or for different variation in size and different variations in various traits, in the environment. So moving on to this acorn experiment. So a little bit of background on growth rates. acorns. So you can have growth in one dimension, you can have it in 2 dimensions, or you can have it in 3 dimensions. So one dimension, let's say if it's just growing vertically, it'll get elongated on the y-axis, so make like kind of like an oval shape. Likewise, if you have it in 2 dimensions, so I have these arrows signifying the left and right. So if it's growing more on the left side than the right side, you'll have kind of like this egg shape, this asymmetric kind of egg shape. And then if it grows in 3 dimensions and the proportions are equal, then it's just going to get larger. So when the shape proportions change, so for instance in 1 and 2, then that's what we call allometric. And these will have, so allometric will have a curve like the power, kind of like an exponential, where isometric, the shape proportions are maintained, so it's a linear relationship. Does that make sense, everyone? So allometric, the proportions are changing. They're not equal. Isometric, the proportions stay the same. So it's essentially just getting larger, but all the dimensions are of the same proportions.
Hey, Sean. So, sorry to interrupt, but with the also thinking linearly, you can have isometric growth in one direction as long as, like, for instance, length and width are growing at the same rate at the same time, and you show that they are linear. But sometimes when you have 3-dimensional growth, you can grow longer in the length than you can in the width, so that results in differential growth instead of equal growth. And then that's when you'll have a more of a power curve, because you can be growing linearly in growth, but you can be growing exponentially in width. So then that would result in that power curve that you see. So, if that makes any sense.
So it's all about the rates of growth, as well as what dimensions it's growing in. So that'll affect whether it's an isometric growth or an allometric growth. So what are some biological reasons why an acorn might grow in 1 dimension, 2 dimensions, or 3 dimensions? This is kind of like some food for thought when you go to write your assignment. This is not really like a right or wrong answer, just kind of brainstorming. So I feel like the obvious one is, like, if you think about an acorn, most of it is an endosperm, or like a food source for the seedling. So if it grows in size, like, you can fit more food for the seedling. You might be giving that seedling a better chance of growing into a tree. But why do you think that it might only grow in like 1 dimension or 2 dimensions, or like acorns don't have like a uniform kind of size? So they have these odd shapes to it. Why do you think that would be? Just like any kind of like biological reason. Maybe it's sturdier, maybe if they're, and I don't know the answer to this, like, for sure. I don't know the answer to this, but I was just like kind of brainstorming. It's like, well, maybe it has this size to attract, birds to pick it up and move it further away, or maybe it like deters predators from eating the acorns, or maybe it attracts predators to eat the acorns and move it to another place. Also, the sizes of the acorns do vary quite a bit. So as you generate your data, looking at like 2 like this, most of these acorns came from the same tree. So why do you think there'd be 2 like extreme differences in sizes between 2 acorns in a set?
Depends on the energy available in that section of the tree, right? Or like how much is being invested. Maybe you think that something closer to the top that has more sunlight, you might be able to invest more energy in a seed there than something that's shaded or on a smaller branch. Or
Yeah,
you know.
absolutely. That's the biggest thing is that the energy allocation. So how much energy the tree is willing to put in toward the reproduction. Also, maybe, the timing of when the acorns were pollinated. So maybe if it was earlier in the season, the acorn might grow larger than, if it was later in the season. If the plant, is water stressed or some kind of stress and doesn't have the energy to allocate toward it, you might see smaller acorns. Just all these different factors are just kind of playing into it. That's why we kind of see that natural variation, we do with acorns. So a couple of potentially useful information. So these acorns were from red oaks. So they're pollinated about 1 year ago. So if you collected them on campus, and that was about a year ago, last spring. You got them from the bins. Some of those were from JT's deck. So those typically had like a much greater range. And he was explaining that some of the acorns were aborted for the tree to conserve energy. So when you see like these tiny little acorns, you refer to these as like aborted acorns. So the tree was having some kind of water stress and just kind of gave up on putting energy into those acorns. And the various sizes of the acorns could indicate when they were pollinated, so at various times throughout the year. So again, if it was pollinated earlier in the season, you might expect to see larger acorns. If it was later in the season, you might expect to see smaller acorns. So does anyone have any questions? So if you guys want to turn to the lab report. So I was just going